A picornavirus is a virus belonging to the family Picornaviridae. Picornaviridae is a family of viruses in the order Picornavirales. Vertebrates, including humans, serve as natural hosts. There are currently 50 species in this family, divided among 29 genera. Diseases associated with this family include paralysis, summer cold, meningitis, diarrhea caused by Enteroviruses, foot and mouth disease caused by Aphthoviruses, myocarditis caused by Cardioviruses, common cold caused by Rhinoviruses, and hepatitis caused by Hepatoviruses. The name is derived from pico, meaning small, and RNA, referring to the ribonucleic acid genome. Thus, pico-rna-virus literally means small RNA virus. Diseases: Enteroviruses infect the enteric tract, which is reflected in their name. On the other hand, Rhinoviruses infect primarily the nose and the throat. Enteroviruses replicate at 37 degrees Celsius, whereas Rhinoviruses grow better at 33 degrees Celsius, as this is the lower temperature of the nose. Enteroviruses are stable under acid conditions and thus they are able to survive exposure to gastric acid. In contrast, Rhinoviruses are acid-labile and that is the reason why rhinovirus infections are restricted to the nose and throat. Taxonomy, Group, ssRNA, plus. Virology. Structure equals, Picornaviruses are non-enveloped, with an icosahedral capsid. The capsid is an arrangement of 60 protomers in a tightly packed icosahedral structure. Each protomer consists of 4 polypeptides known as VP1, 2, 3 and 4. VP2 and VP4 polypeptides originate from one protomer known as VPO that is cleaved to give the different capsid components. The icosahedral is said to have a triangulation number of 3, this means that in the icosahedral structure each of the 60 triangles that make up the capsid are split into 3 little triangles with a subunit on the corner. Depending on the type and degree of dehydration, the viral particle is around 27 to 30 nanometers in diameter. The viral genome is around 2500 nanometers in length, so we can therefore conclude that it must be tightly packaged within the capsid along with substances such as sodium ions in order to cancel out the negative charges on the RNA caused by the phosphate groups. Genome Picornaviruses are classed under Baltimore's viral classification system as group 4 viruses as they contain a single-stranded, positive sense RNA genome. Their genome ranges between 7.1 and 8.9 kilobytes in length. Like most positive sense RNA genomes, the genetic material alone is infectious. Although substantially less virulent than if contained within the viral particle, the RNA can have increased infectivity when transfected into cells. The genome RNA is unusual because it has a protein on the 5' end that is used as a primer for transcription by RNA polymerase. The genome is non-segmented and positive sense. Unlike mammalian mRNA, Picornaviruses do not have a 5' cap but a virally encoded protein known as VPg. However, like mammalian mRNA, the genome does have a poly-A tail at the 3' end. There is an untranslated region at both ends of the Picornavirus genome. The 5' UTR is usually longer, being around 500 to 1200 nucleotides in length, compared to that of the 3' UTR, which is around 30 to 650 nt. It is thought that the 5' UTR is important in translation and the 3' in negative strand synthesis. However, the 5' end may also have a role to play in virulence of the virus. The rest of the genome encodes structural proteins at the 5' end and non-structural proteins at the 3' end in a single polyprotein. The polyprotein is organized as follows: L1ABCD, 2ABC, 3ABCD, with each letter representing a protein. However, there are variations to this layout. The 1A, 1B, 1C, and 1D proteins are the capsid proteins VP4, VP2, VP3, and VP1, respectively. VPO is cleaved into VP2 and VP4 during virion maturation, after the capsid has been formed. The L, 2A, and 3C proteins are proteinases that internally cleave the polyprotein, but these roles vary between genera of Picornaviruses. The 2b, 2c, and 3a proteins interfere with host cell function. The 3b protein is the VPg protein. The 3d protein is the RNA polymerase. Replication equals, the viral particle binds to cell surface receptors. This causes a conformational change in the viral capsid proteins, and myristic acid are released. These acids form a pore in the cell membrane through which RNA is injected, too. Once inside the cell, the RNA uncoats and the strand RNA genome is replicated through a double-stranded RNA intermediate that is formed using viral RDRP. Translation by host cell ribosomes is not initiated by a 5' GCAP as usual, but rather is initiated by an IRES. The viral life cycle is very rapid, with the whole process of replication being completed on average within 8 hours. However, as little as 30 minutes after initial infection, cell protein synthesis declines to almost zero output to essentially the macromolecular synthesis of cell proteins is a shut off a. Over the next 2 hours there is a loss of margination of chromatin and homogeneity in the nucleus, before the viral proteins start to be synthesized and a vacuole appears in the cytoplasm close to the nucleus that gradually starts to spread as the time after infection reaches around 3 hours. After this time the cell plasma membrane becomes permeable. At 4-6 hours the virus particles assemble, and can sometimes be seen in the cytoplasm. At around 8 hours the cell is effectively dead and lyses to release the viral particles. Experimental data from single-step growth curve-like experiments have allowed scientists to look at the replication of the Picornaviruses in great detail. The whole of replication occurs within the host cell cytoplasm, and infection can even happen in cells that do not contain a nucleus and those treated with actinomycin D. Translation takes place by -1 ribosomal frame shifting, viral initiation, and ribosomal skipping. The virus exits the host cell by lysis, and viroporins. Vertebrates serve as the natural host. Transmission routes are fecal-oral, contact, ingestion, and airborne particles. VPg, a VPg primer mechanism is utilized by the Picornavirus, additional virus groups and Picornavirus-like supergroup of RNA viruses. The mechanism has been best studied for the Enteroviruses as well as for the Aphthovirus, an animal pathogen causing foot-and-mouth disease. In this group, primer-dependent RNA synthesis utilizes a small 22-25 amino acid long viral protein linked to the genome to initiate polymerase activity, where the primer is covalently bound to the 5-1 registered trademark end of the RNA template. The uridylylation occurs at a tyrosine residue at the third position of the VPG. A cyactin replication element, which is a RNA stem loop structure, serves as a template for the uridylylation of VPG, resulting in the synthesis of VPG-PUPO. Mutations within the CRE-RNA structure prevent VPG uridylylation, and mutations within the VPG sequence can severely diminish RDRP catalytic activity. While the tyrosine hydroxyl of VPG can prime negative strand RNA synthesis in a CRE and VPG-PUPO independent manner, CRE-dependent VPG-PUPO synthesis is absolutely required for positive strand RNA synthesis. CRE-dependent VPG uridylylation lowers the CMA of UTP required for viral RNA replication and CRE-dependent VPG-PUPO synthesis, and is required for efficient negative strand RNA synthesis, especially when UTP concentrations are limiting. The VPG-PUPO primer is transferred to the 3-1 registered trademark end of the RNA template for elongation, which can continue by addition of nucleotide bases by a DRP. Partial crystal structures for VPGs of foot-and-mouth disease virus and Coxsackie virus B3 suggest that there may be 2 sites on the viral polymerase for the small VPGs of the Picornaviruses. NMR solution structures of Poliovirus VPG and VPG-PU show that uridylylation stabilizes the structure of the VPG, which is otherwise quite flexible in solution. The second site may be used for uridylylation, after which the VPG-PU can initiate RNA synthesis. it should be noted that the VPg primers of caliciviruses, whose structures are only beginning to be revealed, are much larger than those of the Picornaviruses. Mechanisms for uridylation and priming may be quite different in all of these groups. VPg uridylation may include the use of precursor proteins, allowing for the determination of a possible mechanism for the location of the diuredylated, VPg-containing precursor at the 3A1 registered trademark end of plus or minus strand RNA for production of full-length RNA. Determinants of VPg uridylation efficiency suggest formation and/or collapse or release of the uridylated product as the rate-limiting step in vitro depending upon the VPg donor employed. Precursor proteins also have an effect on VPG-CRE specificity and stability. The upper RNA stem loop, to which VPG binds, has a significant impact on both retention and recruitment of VPG and Pol. The stem loop of CRE will partially unwind, allowing the precursor components to bind and recruit VPG and Pol4. The CRE loop has a defined consensus sequence to which the initiation components bind, however. There is no consensus sequence for the supporting stem, which suggests that only the structural stability of the CRE is important. Assembly and organization of the Picornavirus VPG ribonucleoprotein complex. Step 1. 2 3CD molecules bind to CRE with the 3C domains contacting the upper stem and the 3D domains contacting the lower stem. Step 2. The 3C dimer opens the RNA stem by forming a more stable interaction with single strands forming the stem. Step 3. 3D POL is recruited to and retained in this complex by a physical interaction between the back of the thumb subdomain of 3D POL and a surface of one or both 3C subdomains of 3CD. VPG may also play an important role in specific recognition of viral genome by movement protein. Movement proteins are non-structural proteins encoded by many, if not all, plant viruses to enable their movement from one infected cell to neighboring cells. MP and VPG interact to provide specificity for the transport of viral RNA from cell to cell. To fulfill energy requirements, MP also interacts with P10, which is a cellular ATPase. History: in 1897, foot and mouth disease virus, the first animal virus, was discovered. FMDV is the prototypic member of the Aphthovirus genus in the Picornaviridae family. The plaque assay was developed using poliovirus. The discovery of viral replication in culture was also with poliovirus in 1949. Polyprotein synthesis, internal ribosome entry sites, and uncapped mRNA were all discovered by studying poliovirus-infected cells, and a poliovirus clone was the first infectious DNA clone made of an RNA virus in animals. Along with rhinovirus, poliovirus was the first animal virus to have its structure determined by X-ray crystallography. RNA-dependent RNA polymerase was discovered in meningitis, a genus of Picornaviruses. Related families: within the order Picornavirales, there are related viral families, such as the plant-infecting Secaviridae, and the insect-infecting Dicystroviridae. Secaviridae The plant-infecting Picorna-like viruses have a number of properties that are distinct from the animal viruses. They have been classified into the family Secaviridae containing the subfamily Comavirinae, and genera Sequivirus, Wakevirus, Cherivirus, Sagittavirus, and Torradovirus. Dicystroviridae A number of Picorna-like viruses have been described infecting insects. These include Perinanude, the Picorna-like virus of the tussock moth, infectious Flacheri-virus of the silkworm, and Sickbrad-virus of the honeybee, Plowshare Starley intestine virus, kelp fly virus, Ictropioblica Picorna-like virus, deformed wing virus, acute bee paralysis virus, Drosophila C virus, Rhopalosiform podivirus, and Haematobipe virus. Several have been placed in a separate familia the Dicystroviridae. Others have been placed into a new family of Flaviridae. This family includes infectious Flacheri-virus and CIV1 virus. Another virus is norovirus from Drosophila melanogaster. This latter virus awaits further classification. See also, Picornavirales, VPg, animal viruses. References. Additional material, Picornaviridae. Medical subject headings. National Library of Medicine. Retrieved September 3, 2007. Picornavirus. The Bright Institute. Retrieved September 3, 2007. Bar 1 Quarter Chen Osmond, C. ed. ICTVD bar Euro The Universal Virus Database, version 4. Columbia University, New York, USA. Kahn, Cynthia M. Line, Scott, eds. Porcine enteroviral encephalomyelitis. The Merck Veterinary Manual. Merck. ISBN 0-911910-50-6. External links, ICTV International Committee on Taxonomy of Viruses Master Species Lists, ICTV International Committee on Taxonomy of Viruses, Picornaviruses Euro Description, Replication, Disease, Picornaviruses in NCBI Taxonomy Browser, Picornaviridae Classification by the International Committee on Taxonomy of Viruses, Animal Viruses, Virals UN, Picornaviridae, Virus Pathogen Database and Analysis Resource, Picornaviridae, ICTV.